thank you for joining us for our webinar, Why is Understanding the Ecology of eDNA So Crucial? My name is Mario Thomas, co-founder and the CEO of Precision Biomonitoring, and your host today. Joining me for this webinar is Dr. Stephen Crooks, co-founder and head of development at our company. Please note that if during the seminar you lose a sound or image, you should refresh your screen to solve the issue. Also, make sure you close all other programs and apps that you may have opened on your device for better reception. As we all know, environmental DNA, or eDNA, is a relatively new science that has started to revolutionize industry's approaches to biomonitoring. What we are presenting today is a fundamental aspect of these approaches that offer disruptive benefits in accuracy of results and time and cost savings. We appreciate this opportunity to discuss how eDNA can benefit you and have strived to keep this webinar concise and practical. I wish to express our sincere thanks to Stantec, ACOM, Azimut, Parks Canada, Ecometrics, Smithroot, Biomeme, and all the collaborators who have helped us build and validate this exceptional platform developed at University of Guelph in Ontario, and to launch it commercially last year. Our agenda for this morning is a 25-minute presentation, followed by a question period of 20 minutes. It will provide answers to the following three main questions. What are the interactions between eDNA and its environment? How the ecology of eDNA impacts its probability of detection, and why a better understanding of the ecology of eDNA can expand the use of eDNA in the future. At this point, I will invite our co-host, Steve Crooks, to dive into the most interesting aspects of our three topics. Steve.
Thanks, Mario. The subject of this presentation is to reveal the hidden world of environmental DNA, how and from where it is sourced, how it is distributed within aquatic ecosystems, and what fate befalls it in aqueous environments. Following on from this discussion of the ecology of eDNA, we shall explore how the various processes affecting eDNA presence and distribution within habitats impact upon its detection. Finally, we outline future pathways to improve its detection by further refining our knowledge of the ecology of eDNA, which fundamentally underpins its detection and downstream data interpretation. The first step to understanding eDNA as a mark of a species' presence is to define it. We have previously discussed what eDNA is in a previous webinar in what we call a broad sense definition, Sensu, Thompson, and Willerslev. At this broad level, eDNA is genetic material obtained directly from environmental samples without any obvious signs of biological source material. However, we have come to define four narrow sense definitions, which reveal some nature of the processes that act to generate an eDNA signal. These are viable endogenous eDNA, non-viable endogenous eDNA, non-viable exogenous eDNA, and finally, viable exogenous eDNA. Viable endogenous eDNA is DNA that is actively and locally shared from a living organism present at the site of occupancy. This is the eDNA signal that we are all interested in capturing to determine species presence. eDNA is generated as a byproduct of a range of physiological and ecological processes, including the sloughing off of cells from gut and cloacal walls, which may be deposited during urination and or defecation. There may also be a turnover of cellular material in the skin of certain animals, such that dead or replaced cells are deposited in the immediate environment. Other potential sources include the regurgitation of foodstuffs and cells contained within, and through the exudation of mucus and external signaling pathways in the skin. Perhaps the most significant source of eDNA is through the release of gametes, particularly important to detect those animals with external reproduction. eDNA may also be released as a result of the biological breakdown of once living organisms through the processes of decay and decomposition, which we call non-viable endogenous eDNA. As the organism is releasing eDNA as it decays, this signal is local, but is no longer indicative of a living animal or plant. For detection purposes, this signal is still telling of a local population. After all, everything dies, but the eDNA signal from viable and non-viable sources would conflate their provenance, complicating inferences of local population abundance or biomass. There is also a secondary source of non-viable endogenous eDNA, and that's from predation of a target organism by in situ predators, who then go on to deposit the target's eDNA in their own feces. Good content analysis of predators has established that fecal DNA analysis can identify those species predated upon by top-level consumers. Therefore, this seems to be a not insignificant potential source of non-viable eDNA. Our third and fourth narrow-sense definitions describe potential sources of target eDNA that may not reflect local population shedding. These are potential sources of exogenous, also called allochthonous, eDNA. The first of these definitions is viable exogenous eDNA, whereby the nature of the aquatic system may redistribute eDNA that is locally emitted away from its site of shedding to some other potentially distant sites where it is subsequently sampled and then detected. One obvious potential hydrological influence in this redistribution of eDNA is water flow in riverine systems, whereby high water flows can transport eDNA distances downstream. Our last narrow sense definition is of non-viable exogenous eDNA, in which our target organism is consumed by a predator at the site of its true occurrence, but then that predator deposits the target's eDNA in some other distant water body. Very few studies have looked into how significant a source this is, but its possible existence should be kept in mind. What is the fate of eDNA once it is exposed to the local environment? That will depend on the biotic and abiotic characteristics of the local environment. The fate of all things is to decay eventually, and therefore eDNA is no different. It may be released as whole genomes, still temporarily intact within cells, or as large fragments post-digestion, but ultimately many factors will act in synthesis or in sequence to break down cellular and nucleic acids beyond detectability. We've already touched on the fact that it may be removed from a site and whisked away via downstream flow, for example. But if a strong signal remains local, as is expected as the target will continually emit eDNA, what factors influence its breakdown? Increasing research efforts in the academic realm has sought to determine the major sources of eDNA decay and sequestration, and which environmental metadata are correlated with higher or lower than expected detections of target organisms. Intuitively, many investigators turn to the usual suspects of chemical and biotic decomposition: pH, ultraviolet light, temperature, levels of dissolved oxygen, levels of substrates within the water, biotic filtration, and finally, microbial digestion. Depending upon these conditions, eDNA may persist for as long as months or be gone in mere days. pH is an obvious target, as anybody who works in molecular biology can attest. Deviations from a specific pH can alter the chemical constitution of macromolecules and alter the parameters of chemical reactions. Very low pHs, under 5, or very high pHs, greater than 9, can destabilize double-stranded DNA, making it more amenable to digestion. Also, low pH can cause the depurination of DNA, but also increase its insolubility. Low pH has often been correlated with reduced eDNA detection in both mesocosm and wild surveys. Low pH not only affects the physical structure of DNA, but can promote the digestion of DNA into mononucleotide subunits by the action of microbial enzymes, including those produced by widespread fungi and bacteria, for example, Aspergillus and Streptomyces. However, more neutral pH conditions are conducive to large microbial populations, which may digest eDNA through the exudation of nucleases, a natural process in microbial biology whereby DNA is cut and potentially even interred into microbial genomes through a process of lateral gene transfer. There is a slight and probably minute but yet unquantified possibility that the eDNA fragment of a target organism may be taken up by microbes and artificially amplified through bacterial binary fission. Whilst this may seem fanciful, the uptake of woolly mammoth ancient DNA has been shown to occur in bacteria exposed to DNA from 43,000-year-old bones. Levels of nutrients can alter microbial communities, which in turn may have cascading effects upon eDNA uptake. For example, it may be used as a source of phosphorus for some microbes. Oxygen levels are an obvious limiting factor on biotic communities, and eDNA degradation was observed to be greatest when oxygen demand in a system was also elevated. Low oxygen content sediments are prime repositories of eDNA, in which it may persist much longer in these stable conditions than when exposed to the water column above. Fresh waters are characterized by their lack or much lower amounts of dissolved salts. Salinity may have an effect on retention of eDNA or its solubility in submarine, brackish, or estuarine conditions. Elevated salt levels may precipitate free DNA out of solution from the water column, undermining detection. Collins and colleagues in a paper published last year found eDNA to decay 1.8 times faster in the inshore region versus outshore along a temperature salinity gradient, with an estimated detection window of 48 hours in the inshore region. However, this likely reflects greater biotic activity associated with the nearshore area. DNA will break down in the presence of strong ultraviolet light. However, there is little evidence so far that solar levels are strong enough to impact eDNA decay levels noticeably more than any other factor. It is also not clear how other biotic processes, such as filter feeding by mussels, will act to remove eDNA-containing particles from water bodies. Makler and colleagues found no evidence for UV-assisted breakdown of eDNA or of the presence of the zebra mussel affecting detection rates, at least in 3 aquatic macroinvertebrates in a mesocosm experiment. If unaccounted for, eDNA will break down quite quickly, perhaps between the shedding event and detection event, and as a result, may not be detected. Therefore, there is a not insignificant risk of false negative detection in areas prone to high levels of eDNA decay. It is impossible to remedy this situation entirely. At PBI, we can mitigate for the presence of factors linked to reduced eDNA detection by performing a priori surveys of aquatic ecosystems, usually in the form of pilot surveys. As a result, subsequent surveys are optimized to sample in areas likely to have high eDNA retention rates. Further, as another guard against decay, our on-site eDNA extraction kits instantly preserve the DNA in a thermostable and pH-stable elution buffer. It is expected that eDNA within a system is not likely to be homogeneously distributed. Benthic animals will likely deposit eDNA directly on the benthos, whilst midwater or surface inhabiting animals are likely to deposit in the water column. Deposition of eDNA on the benthos may leach down to become commingled with conserved and more historic eDNA molecules, which we here call sedimentary eDNA to distinguish from contemporary eDNA, such that recent and historical signals become conflated if too much sediment is sampled for eDNA extraction. The ecology of the organism is paramount in understanding the distribution of a target's eDNA in a system, as diel and seasonal movements are expected to vary significantly within and among highly motile and sedimentary animals, including the variant depths that some animals move up and down the water column during their daily travails. Notably, spawning migrations can result in the targets moving long distances from their pre-spawning and otherwise regular haunts, so that a big spike in eDNA deposition in the form of gametes may take place kilometers from their usual sites of occupancy. At PBI, we employ sampling schemes to maximize the detection of contemporary eDNA, for example, largely avoiding sediment samples for pelagic or open water species, although this is dependent upon the habitat preferences of a target within the water column. All species are different in how, where, and when they will shed eDNA. Physiological eDNA shedding rates are likely to be linked to metabolic rates and food consumption, activity levels, and the release of gametes. Some taxa undergo metabolic depression and exhibit significant seasonality. Freshwater turtles will leave the water during the nesting season, and even when they overwinter under the ice, their inherent shedding rate may be significantly downregulated. Territoriality and intraspecific migrations can be commonplace within some species, with concomitant changes in habitat preferences and even physiological and anatomical traits, for example, as exhibited by anadromous salmonids. Highly motile species might tend to shed eDNA over greater distance, whereas sedimentary species may release only very localized eDNA emissions, the fate of which might depend more on hydrological features or whether they're exposed to the water column or whether they are fully or partially buried in the benthos, for example, unionid mussels. Of course, many species undergo long-distance migrations for spawning purposes, and knowing the location of its habitat is crucial for monitoring. However, external spawning might increase detection due to the high-intensity plumes of gamete release. A working knowledge of the biology of the target organism is absolutely essential from upfront assay design, for which the genetic characterization and distributions of populations and conspecifics is paramount, to conducting field validations and client surveys. At PBI, we pride ourselves on applying this breadth of knowledge to maximize the probability of detecting the targets when it is at a low abundance. As responsible eDNA practitioners, we need to justify the use and deployment of a particular assay in any given aquatic ecosystem, lest the technique not be adopted by regulators, clients, end users, and managers in aquatic biodiversity conservation. One of our primary building blocks in this regard is a thorough analysis of assays in field pilot surveys, which is a recurring theme in this presentation. Having been released, cells or free eDNA are at the mercy of significant hydrological forces. Shogren and colleagues in 2018 developed a basic conceptual model of transport and retention of common carp eDNA in a model stream system, finding that substrate types vary in their eDNA retention times, with turbulence also contributing to the variance of eDNA deposition, retention, and release. However, more study of the 3D distribution in space and time of both particulate eDNA and free molecular DNA molecules needs to be conducted in all possible habitats, lentic, lotic, tidal, and open ocean. Likewise, with species ecologies, top-end eDNA practitioners should be fully cognizant of the hydrological systems and features within which they will be sampling. Not accounting for variation within and among water bodies, seasons, and sampling years will undermine effective and reliable eDNA surveys. At PBI, all of our species pilot surveys are actually portfolios of studies encompassing different watersheds, hydrologies, and geologies of substrates, which may impact PCR inhibition, for example. But on this, we'll discuss more later. More fundamentally, we may need to tailor the pore size and water volumes to be extracted from a site. All of these can be modeled with effective pilot study design. Both cellular material and free DNA will break down, and if a source is removed, it will be beyond detectability in days in some environments. Potentially rapid decay suggests that a detection by PCR of a water sample reflects a recent shedding event and can be construed with some confidence that the deposition of that molecule was made within hours or days. However, there is a further barrier to detection caused by environmental factors, and that is the process of PCR inhibition, wrought by the binding of certain chemicals to DNA, negatively impacting the performance of PCR by interfering with reagents during the reaction. eDNA binds to organic matter. This advection to particulates is a part of the story of its distribution in the system, as previously discussed. But such particles are often heavy with organic acids, such as humic acid, produced as a byproduct of decay and tissue disintegration. Humic acid binds to DNA in ways that may inhibit downstream PCR analysis. There are other sources of PCR inhibition in the environment, including, but not limited to, complex polysaccharides, biosalts, and urea, all of which may be deposited into the environment through biotic waste removal pathways. Other inhibitors include tannic acids, phenols, proteinases, and molecular sources of calcium ions. The distribution of inhibitors, much like eDNA, is heterogeneous and therefore must be accounted for in all eDNA analyses, as this will be discussed later. Testing for PCR inhibition is absolutely crucial, as not accounting for it will result in a significant false negative error. This is why all eDNA extracts should be subject to a secondary qPCR analysis performed either simultaneously or in sequence with that assay for the target species. This assay is termed an internal positive control, or IPC, and is constituted of a not found in nature fragment of DNA that is amplified by bespoke primers and a probe as per normal qPCR, except that the fragment is inoculated into every reaction in exactly the same number of copies, for example, 100 copies per reaction. When run in replicates using pure water as a template, or in this case, no template control, we get the exact same CQ value within an acceptable level of variation, as there is no inhibition in pure water. If the eDNA extract contains no inhibitors, then when using this as a template, we should achieve the same CQ value. However, if there is PCR inhibition, then the CQ value will either be higher or completely absent, indicating late or complete inhibition. If inhibition is confirmed, then post-extraction processing of the sample is necessary, or innovations found to nip the problem in the bud during the PCR reaction itself. PCR inhibition has to be dealt with. Usually this involves dilution of the sample, adopting commercially available environmental master mixers, or using some other cleanup process, for example, spin kits. However, these are frequently ineffective or even counterproductive, as cleanups can often drastically reduce eDNA yield, which is problematic for those species that have very low levels of eDNA in any possible extract. Here at PBI, we are assisting in the development of a PCR inhibition attenuation process by developing a brand new master mix that negates the effects of PCR inhibition from samples collected from the local Speed and Eramosa rivers in southern Ontario. Early trials have a 100% recovery rate of PCR detection using our IPC as the experimental target. We are further refining this work and performing more tests on IPC and naturally occurring eDNA and across multiple sites of variant water qualities. We have just reviewed the major sources of eDNA, how it is broken down, and how it might be distributed in a system. The net effect of all these processes is a dynamic system of eDNA molecules in constant flux and with both deterministic and stochastic processes at play. Therefore, optimizing the detection of a target eDNA is no simple matter, and all of these factors must be accounted for to maximize that detection. Of paramount importance is data, site-specific data generated from pilot studies, so that almost all the factors discussed can be assessed. Ideally, pilot data should encompass the seasonality and biology of a target organism, as it is no good performing pilot sampling within a habitat in which the target is not expected to be yet present. A note on maximizing detection using molecular tests such as qPCR here at PBI. We recall how it is expected that the most common form of a target's eDNA would be in terms of smaller fragments. Therefore, right at the beginning of the assay design process, we immediately increase our chances of successful detection by targeting only very small fragments of eDNA, usually between 70 and 250 base pairs. Targeting small fragments also has the added benefit of increasing the sensitivity of the assay, as amplifying small fragments is more efficient than amplifying bigger ones. Environmental DNA is a phenomenon. It is itself not a tool, but we deploy sampling, statistical, and molecular biological tools to infer the presence of taxa based on the results we obtain. Therefore, it is not a black box. EDA detection as a tool must be wielded in a responsible manner and ideally be replicable and repeatable without losing power to detect the target if it is present. Once we have a fully lab-validated assay, we must deploy it in the field to ascertain its utility. The bottom line is that all results must be biologically meaningful. The rule of sampling number 1 is that the habitat must be suitable for the species in question. Otherwise, it is folly to expect a meaningful detection. In fact, if detection is observed in an unsuitable habitat, this is some evidence to suggest the presence of exogenous eDNA and should probably be discounted as being local. A keen understanding of the ecology of a target is of utmost importance. However, survey habitat could be theoretically suitable for the target species, even if it is currently not occupied in that location. For example, some habitats are suitable for invasive species that have yet to make a foothold. In such cases, our surveys would still hold validity. Returning to a common theme at PBI and this presentation, pilot studies allow us to validate every aspect of our eDNA workflow. Goldberg and colleagues pointedly emphasized the importance of performing pilot studies. We very much agree with her. Pilots are important for a number of reasons, some of which we have discussed. What we haven't remarked upon yet, but is extremely important, is that the validation of a qPCR assay for use in a study system in which the target is predicted to be extant or have previously been recorded or have a potential to exist needs to be tested. However, the assay has to be ground-truthed in a validation pilot to provide baseline estimates of its power to detect the target. Few power analyses have been undertaken in eDNA studies, but these are of vital importance to estimate minimal effort as influenced by total volume of water sampled, the number of technical replicates in qPCR, or the number of iterative sampling events, for example, days or interseasonally, to be expected to achieve a power of 95%, assuming an error rate of false negative of 0.05. These can be done under idealized conditions, for example, in captive populations or mesocosms, where the target's density is known, but should in all cases be conducted in natural settings with sites of known occupancy to give a non-idealized sampling intensity to achieve the required power. Pilot studies are essential for also measuring some or all of the variables previously discussed that have been implicated in eDNA decay or PCR inhibition. Standard environmental monitoring equipment can now easily measure with precision and accuracy pH, conductivity, sediment load, turbidity, dissolved oxygen, temperature, UVB radiation, and also continuous hydrological features such as flow rate. These parameters can be correlated with eDNA detection success. These data serve as primary input into habitat occupancy models, which can be used to determine site-specific parameters, including the probability of detection, site occupancy, and the probability of detecting the target in any given biological sample. The first aim of pilot studies is to estimate the probability of detection. So the habitat occupancy analysis can tell us if the achieved probability of detection is good enough or if it is too low. The required level of probability is likely to vary for given taxa and be based upon its conservation status. If too low, then we can modify the sampling scheme, increase technical replicate effort, or check for PCR inhibition. Occupancy analysis can tell us which metadata correlate with detection success, for example, informing us if any sites are prone to PCR inhibition, or if the detection of a target is positively associated with any habitat features, for example, the levels of macrophyte coverage, so that sampling can be targeted spatially and with more precision. summary, the better our understanding of the causes and processes that predict the sources, the longevity, and the distribution of eDNA, the better able we are to detect target species for which eDNA is a highly effective proxy for occurrence. At PBI, we focus on small target marker size. We validate assay performance in iterative rounds of pilot trials, encompassing as much of the variation in eDNA predictors as we will face, and record, correlate, and model how effective each factor is in predicting detection success. With every iteration, our knowledge of where, when, and what to sample proceeds incrementally, further refining our assays and surveys. To assist in this endeavor, we have formulated a bespoke sampling design algorithm to optimize sampling based on pilot and environmental data, to standardize the sampling design process for individual and often idiosyncratic projects. To this end, we have just filed a patent with the US Patent Office and will have a prototype ready by the year's end. Further in-house innovations include increasing molecular discrimination of extremely closely related taxa so that we may begin to perform population-specific analyses or to target cryptic species that are otherwise hard to detect using morphology, but are sympatric with overlapping ecological preferences. And to reiterate, we are co-developing a new reformulated master mix to tackle the problem of organic inhibitors present in eDNA extracts.
Thank you very much, Steve, for this very informative presentation. And thank you all for your participation. As we have shown, obtaining reliable eDNA results is a science and a process a lot more sophisticated than a simple grab and go. We now invite you to ask any question you may have by clicking on the question button below the presentation and writing them down in the space provided. Steve or I will strive to answer your questions to the best of our abilities. So we will be taking questions now. So just click on questions, insert your question by typing it in, and I will answer the questions verbally, or Steve will. So who has a question? Here's a question from Nick Hodges from ACOM. What is the status of the Biomeme field analysis equipment, and how do you see effectiveness of that equipment being improved over time? Nick, the answer to this is that we are just in the process of launching with Biomeme a new unit called the Biomeme 3.9, which is a much improved qPCR tool for the field. It has a better architecture, better algorithms, and can run 27 reactions. It has 9 wells and 3 fluorescent channels. So, for example, with an IPC, we can duplex assays and run these tests in triplicate, sextuplicate, or more. And the development work continues. We foresee that over time, the use of on-site qPCR will become better and better. We participated in the testing of the prototype of the 3.9 in several studies, and I've seen a major improvement. And the unit that you know, the 2.3, has been discontinued. So next question by Kenny M. Could you proceed to amplification or labeling steps immediately from water samples, or do you still carry out extraction protocols? If so, do you need specific kits or solvents?
Yeah, that's a straightforward question. What we use on site, we often almost essentially 100% filter the water using filter papers in single-use cartridges, so there's no chance of contamination from improper bleaching. And then we use on-site eDNA extraction kits produced by a company called Biomine, and we perform DNA extraction on-site, so the DNA is immediately extracted from the filter paper. That means there will be no degradation in transport from the site of detection back to a home lab where PCR will be performed, or indeed we can perform PCR in the field with the 3.9 device.
A question from France Brunel. Can we trace the whole path of a pathogenic insect in an agricultural field or a city with the eDNA? Can we trace the whole path of a pathogenic insect in an agricultural field or city with the eDNA?
Yeah, I think that's possible. If I understand you correctly, you want to trace the source of infestation and track its progress in an agricultural setting. I think we can. We have worked with people at NRCan, where we've used the Biomine extraction kits to extract DNA from insect legs. It's been modified for use with thermocyclers, portable thermocyclers we use. I think it's very possible with the correct sampling design that we can trace the spread of any pathogen, be it aquatic or in this case within an entomological setting.
Next question, please. Is there any other question? I realize that we are near the end of. Oh, here's a question from Tanya Valdivia. So Tanya's question is: in a prospective study, given the variety of conditions that can alter the presence detection of the eDNA, how do you decide when is a good time to perform the sampling?
That is a very good question. Of course, it depends when you want to actually perform the survey proper. If you're looking at sporting sites, for example, you're looking at where a fish may choose habitat in which to spawn. Ideally, for a pilot study, we would need to do a pilot in a previous spawning run to make sure we can identify eDNA hotspots associated with gamete effusion, if you would. And then we would measure all aspects of the environmental metadata and the flow rate. Of course, some of these things vary from season to season, from year to year. For example, flow rate is dependent on rainfall and all sorts of other factors, deforestation. So some things are very hard to model, but by modeling as best as we can from previous seasons, from a previous season, you're in a better shape in the following season to perform a much more rigorous survey.
Thank you, Steve. Is there any other question that we can address on the topic of eDNA ecology this morning? All right, so I think that we have answered all the questions that were posted. Again, I want to thank you for participating in this webinar Our next webinar will be in about 4 or 5 weeks. You will receive an invitation. The topic of the next webinar will be practical applications of environmental DNA and the case studies that have been performed with our collaborators. In the meantime, if you have eDNA survey projects that are coming up, don't hesitate to contact us, precisionbiomonitoring.com. Our contact information is on the website, and we'll be delighted to meet with you, have a conference call, and discuss your project and see if we can help you obtain more reliable results from your eDNA survey. So thank you very much. Again, thank you, Steve, for this presentation. And until next month, when we will have our next webinar. Enjoy the rest of the day. Bye-bye.